Friends, welcome to Biology Made Easy. We are discussing the different evidences of evolution, and today in this video we will discuss embryological evidences, or you can tell evidences from embryology. Embryological evidence. So first we have to know what is embryology. Embryology means the early development in an organism. That is known as embryology. If we will make a comparison between the development of embryos of different animals, we will find that there is a striking resemblance between the different organisms. What is the reason behind it? So the probable reason is that all of them have been originated from a single ancestor. That is why during their early stages of development, during their embryo journey, they show similarities. So let us give examples one after another so that our understanding will be better. And the first example we will take is from the resemblance in early development. Resemblance in early development. Now, usually in all animals, the starting point is the zygote. That means when one egg and one male gamete combine together, the zygote is formed. So the zygote is the starting point for all organisms. Now this zygote undergoes cleavage. It undergoes cleavage and it gives rise to morula. And the morula is completely a solid structure. Now this morula again it divides and gives rise to blastula, which is a hollow structure. Then the blastula again divides and it gives rise to gastrula. Now this gastrula is three-layered, and from the three layers is one is ectoderm, the second is mesoderm, and third is endoderm. In case of all animals, the different organs they develop only from these three layers, and in all organisms this same organ develops from the same layer, and this is the stages of these are the different stages of development. So it means that those organisms which pass through these processes, they have been evolved from the same ancestor. That is the evidence from embryology. Now come to the second point. The second point is resemblance in, this is similarities in early development, and this is resemblance in early development. Now if we compare the embryology of fish, salamander, tortoise, chick, and man, and suppose we will study in different stages of development. Suppose after second month, fourth month, or sixth or seventh month, like that, if we will compare the embryology of a fish, salamander, tortoise, chick, and man, we will see that the embryological development is similar in all cases. That means the evolution has taken place in this way. They are from the fish to salamander, tortoise, chick, and man. So this is one of the great evidences, that is similarities between resemblance in the early development. Then the third one is still more important, that is known as recapitulation theory. recapitulation theory. In 1828, one scientist known as von Baer, he proposed a law known as Baers law, according to his name. But in 1866, another scientist, Ernst Haeckel, he modified this Baers law into biogenetic law. Biogenetic law. And what is the meaning of this biogenetic law? It is very important for examination point of view. Biogenetic law means ontogeny repeats phylogeny. Ontogeny repeats phylogeny. So this is very important for examination point. Now what is the meaning of ontogeny? Ontogeny means the life history of an organism, and phylogeny means the evolutionary history. So ontogeny repeats phylogeny. That means during the early development of an organism, it recapitulates the ancestor. That means from which it has been evolved. So we can better understand it in a better way if we give some examples. So let us give one example from that of the fish, or we can better give that of the frog. Now in case of frog, there is the larva stage is known as tadpole larva. And the tadpole larva, it is just like fish-like, and it has tail fins for swimming, and gills for breathing. That means the tadpole is one of the stages, one of the embryological stages of a frog, which looks fish-like, having tail fins which help in swimming and gills for breathing. But in case of adult frog, these are not there. So it indicates that the frog has been evolved from fish, from the aquatic organisms. This is one example. Now the second example we can take from that of the from the plant we can take, that is protonema of moss and fern. Protonema of moss and fern. So in case of moss and fern, when the spore germinates, it gives rise to a green branched filamentous structure which looks like a filamentous algae. That means it gives a structure like this, a branched structure. It gives like a structure like this. So this is known as protonema, and this is the early stage of development in case of moss and fern, but it is to one of the filamentous alga. That means the bryophytes, the mosses, and some of the ferns, they have been evolved from the aquatic alga. That is one of the examples. And another example also we can give from that of the gymnosperms. So usually the gymnosperms, they do not require water for fertilization. Do not require water for fertilization, because their gametes are non-motile, and usually the fertilization is siphonogamy. They don't require, they just pass through the pollen tube. But in case of a primitive gymnosperm like cycas and ginkgo, they have ciliated sperms. Ciliated sperms, or you can tell they have their sperms are flagellated. So this flagellated sperm is characteristics of the lower ones, that is bryophytes and pteridophytes, which require water for fertilization. So in the same way, in case of cyclos and gingo, they require water for fertilization, while the higher gymnosperms, they don't require water for fertilization. So this is another good example from that of the gymnosperms. And another example is that of acacia seedling. Acacia seedling. Acacia seedling, during the early stage of development, it has simple leaves. Simple leaf. But in case of adult acacia, it has compound leaves. That means the plant has been developed from some other plants having simple leaves. So this is the examples from that is recapitulation theory. How in the early stages of development, how some organisms they resemble their ancestors. Now let us discuss the fourth point. The fourth point is temporary non-functional embryonic structures. So the fourth point is temporary non-functional embryonic structures. Embryonic structures. Now, what are the examples? You will take the embryo of birds. Embryo of birds. Now they have tooth buds. The embryo of birds have tooth buds, but the tooth buds are they are degenerated. They are lost before hatching. Lost before hatching. That means the birds that do not have teeth. Now another thing is in the embryo of birds there is also mouth, but they simply observe. They observe from yolk through vessels. That means they do not use the mouth parts. Whereas if you will come to the adult birds, the adult birds they require teeth because usually they feed on solids like grains, nuts, meats. So they eat such solids which are very hard substances, but they do not have teeth. So initially in the embryonic stage there are tooth buds that is lost during hatching. They have a mouth. They don't use mouth. They simply absorb the yolk through vessels. But in case of adults, they have mouth but without teeth. Even if they eat some solids like grains, nuts and meats. So it indicates that birds have been evolved from some reptiles having teeth. They have been evolved from reptiles having teeth. Now let us give one fifth example. The fifth example is we can take development of vertebrate organs. That means if we take the case of fish. In case of fish, the heart is to, let us take the case of heart. Because in the development heart, brain, kidney, you will find some similarities. But let us take the case of heart. In case of fish, heart is two-chambered. Two-chambered and one auricle and one ventricle. But in case of the amphibians, it is the three-chambered. Three-chambered. That means two auricles and one ventricle. Now, if we come to the case of reptiles, it is usually, we can tell incompletely four-chambered. That means here also we will get two auricles, but here partly divided ventricle. And if we come to the case of birds and mammals, completely it is four-chambered, having two auricles and two ventricles. So it clearly shows that the birds and mammals, they have evolved in this method of evolution. So this also gives example that the development of certain organs like heart, brain, kidney takes place in a gradual development from lower to higher organisms. Then we can give another example, that is resemblance among vertebrate larvae. In some vertebrates, there is similarity in their larval structure. So one such example, we can take the annelids and mollusks. So in these both cases, their larvae is similar and is known as trochophore. Similarly, if we take the case of echinoderms and hemichordates, in both these cases, they are also larvae is similar larvae. So these are the different evidences from embryology to prove how the evolution has taken place. So this is about the embryological evidence. Now let us discuss the questions. So one question we can take. First question, embryological support for evolution was given by first choice Van Baer, second choice Charles Darwin, third choice Alfred Wallace, and fourth choice Ernst Haeckel. Now the question looks very simple, and actually it is simple, but this question was asked in NEET 2020. Then the second question. Second question is Haeckel's biogenetic law is first choice ontogeny repeats phylogeny. Second choice phylogeny repeats ontogeny. Third choice reproductive isolation. And the fourth choice, every organism is produced by its parents. So you see, these are the type of questions which are usually asked. You see that question was asked only in the NEET 2020. So I hope you have a very clear understanding about the embryological evidence. Thank you very much for watching the video. God bless you.